In this video, we will explore the fascinating field of microbiology and focus on viral pathogenesis. We will discuss topics such as viral attachment, replication, host interactions, immune evasion, vaccine development, antiviral therapies, emerging viruses, and more. Understanding these mechanisms is crucial in our fight against viral diseases. Viruses use specific host cell receptors to attach and enter the cell. They can enter through endocytosis or membrane fusion. Understanding these processes helps develop antiviral strategies to prevent infection and reduce spread. In this section, we will explore how viruses replicate within host cells. We will discuss the different strategies used by viruses, the steps involved in replication, and the importance of understanding this process for the development of antiviral drugs and immune therapeutics. Viruses utilize various strategies to manipulate host cells for survival and replication. For example, they inhibit interferon signaling, hijack cellular mechanisms like protein trafficking and cytoskeletal rearrangements, and exploit host defense mechanisms. Targeting these interactions can aid in developing antiviral interventions. Viruses have evolved various ways to evade the immune system. They can mimic host molecules, interfere with antigen presentation, and inhibit interferon signaling. Understanding these strategies is vital for developing effective treatments and vaccines to combat viral infections. In the fifth section on viral oncogenesis, we delve into how certain viruses can cause cancer. These oncogenic viruses integrate their DNA into our cells, disrupting normal gene function and leading to uncontrolled cell growth and tumor formation. Additionally, viral proteins can mess with cell cycle control, hindering checkpoints and resulting in excessive cell division and tumor formation. Understanding these mechanisms is crucial for developing preventative measures, including vaccines and targeted therapies. Viruses can lead to specific organ damage, causing various diseases. Understanding viral pathogenesis in specific organ systems is crucial for managing infections effectively and developing targeted treatments. Examples include respiratory viruses causing pneumonia and neurotropic viruses affecting the central nervous system. Vaccines are important for preventing viral infections. Different strategies are used in vaccine development, including live attenuated, inactivated, subunit, viral vector-based, and nucleic acid-based vaccines. Each strategy has its strengths and limitations, and understanding them is essential for effective vaccination programs. Examples of vaccines include the MMR vaccine, HPV vaccine, Oxford/AstraZeneca COVID-19 vaccine, and mRNA vaccines. In the antiviral therapies section, we will discuss drugs that target specific stages of the viral life cycle. Nucleoside analogues disrupt viral replication by incorporating into the viral genome. Protease inhibitors block viral enzyme activity, fusion inhibitors prevent viral entry, and polymerase inhibitors interfere with viral genome replication. Neuraminidase inhibitors inhibit the release of viral particles. Understanding these mechanisms is crucial for effective use of antiviral drugs while limiting drug resistance. The emergence of new viral diseases, including those that can infect humans from animals, zoonotic diseases, poses a global health challenge. Understanding their origin, transmission, and potential for causing pandemics is crucial for disease control. Examples of zoonotic diseases include Ebola, MERS, and COVID-19. Genetic changes in existing viruses can also lead to the emergence of new diseases. Studying the ecology, epidemiology, and evolution of these viruses helps in developing early warning systems and effective interventions. Investigating emerging viral diseases improves global preparedness and response to future outbreaks. The genetic makeup of individuals plays a crucial role in determining their vulnerability to viral infections and the subsequent progression of the disease. By studying the relationship between host genetics and viral susceptibility, we can develop targeted interventions and personalized treatments. Genetic variations in genes involved in viral recognition, immune response, and viral replication can impact susceptibility. Research using genome-wide association studies and genetic profiling has identified several host genetic factors associated with viral susceptibility, leading to the development of novel preventive strategies and personalized antiviral treatments. Some viruses, like Epstein-Barr virus, herpes simplex virus, and HIV, can establish long-term infections by entering a dormant state or continuously replicating at low levels. This understanding helps develop strategies to control these infections. Zoonotic viruses, which can jump from animals to humans, are a serious global health concern. Understanding how these viruses interact with their hosts is essential for preventing and managing outbreaks. For example, coronaviruses like SARS-CoV, MERS-CoV, and SARS-CoV-2 likely originated from bats. Research on these interactions involves studying how viruses cross species, replicate, and spread in humans, as well as identifying risk factors for spillover. This knowledge helps in developing surveillance systems, control measures, and strategies to minimize the impact of future outbreaks on human health. Understanding how viruses interact with their hosts is crucial in deciphering infection, immune response, disease progression, organ-specific interactions, and interspecies transmission. Through research, we can develop antiviral therapies, vaccines, and preventive measures against emerging viral diseases. Improving diagnostic tools, surveillance systems, and interventions will promote a healthier future and minimize the impact of viral infections.